Dear students, please do hurry and join the live session so that we can start the live class. Please do hurry and join our live class so that we can start the live session. Before we dive into today's session, make sure to subscribe our YouTube channel, Eduins, if you have not already yet, so that you never miss the live classes and other informational educational videos. Please do hurry and join the live class so that we can start the live session. And this is an announcement for especially for the board student. You can visit our website eduins.in and purchase our premium version at a very low cost, where you can get unlimited study materials, mock test series, and recorded classes. Please do hurry and join the live class so that we can start our live session. A very good evening to all of you and welcome to another live class of Eduins. Today we will discuss about the cell division and cell cycle part 2. In previous class we have discussed the cell division and cell cycle part 1, where we have discussed about the chromosome, its structure and the different parts of the chromosome. Today we will discuss about the actually what is cell division and cell cycle. So what is cell division? In previous class we have already discussed that cell division is the process of generating new cells, that is called daughter cells from the pre-existing one, okay, which is called mother cell. Okay, so this is the cell division. So what is the importance of cell division? Of course, cell division is important for every living organism. Why? Due to growth. Number one, for the development from a child to adult. Okay, so the growth is important. Number two, repair. To repair the cut or wounds of our body. To repair the wounds. Okay, and number three, and most importantly, that is for reproduction. That is continuity of gene flow. Okay, so these are the importance of cell division. importance of cell division. So the cell division is mainly three types. What are they? The types of cell division. Cell division. Number one is amitosis. Number two is mitosis. And number three is meiosis. These are the three kinds of cell division observed in living organism. The first one is amitosis actually occurs in the prokaryotic cells. Prokaryotic cells or prokaryotic organism. Okay. What is prokaryotic? Pro mean old and karyon, that is derived from the word karyotic. Karyon mean nucleus. Okay. So primitive or old type of nucleus have those organisms shows the amitotic or amitosis cell division. What are they? Like bacteria, yeast. Okay, these kinds of primitive organisms shows this type of cell division. Second one is the mitosis, which is also known as the indirect cell division. Okay, and next one is the meiosis, that is called reductional cell division. Okay, so these are the three kinds of cell division. We will discuss it elaborately. First, amitosis. First one is amitosis. Okay, so in amitosis, if this is an organism, of course, most of the primitive organism or prokaryotic organism are unicellular. So if this is a unicellular organism, this is the cell and this is the nucleus. Okay, so the division will be direct division. Like first, these are the genetic materials, and in next step it will divide into two different cells. Okay, this is the mother cell, this is called mother cell, and these two are known as daughter cell. Okay, so this is the process of amitosis. This is actually a simple division, simple or direct division. Okay, so the next one that is very much important for our body, that is called mitosis. Next one is the mitosis. Okay, so this is the an indirect cell division. How? In meiosis, simply for the understanding, that is, it divides from this to this cell. But lots of process involved here that are enlisted this phase. This is called mitosis or mitotic division phase. Okay, so there have 4 phases in mitosis division. Mainly the 2 phases there. Number 1 is karyokinesis and number 2 is cytokinesis. Of course, from the name, that is karyokinesis, kinesis means division, and karyo means karyon, that is division of nucleus, and the cyto means division of cytoplasm. Okay, and this karyokinesis is further divided into 4 phases. First one is the prophase. Second one is the metaphase. Third one is the anaphase. And fourth one is the telophase. Okay. So what changes occurs in these phases? So for this, we will discuss elaborately. But before that, for the cell division or mitotic division, the cell organelles that are responsible for the or help in cell division, number 1 is ribosome, that produce protein. Number 2 is centrosome or microtubule, and number 3 is mitochondria. These 3 cell organelles are very much important for the cell division. Why? Because ribosome synthesizes the protein, centrosome in case of animal cell, and microtubule in case of plant cell are important, and the mitochondria, which is important for the synthesis of ATP or energy. So these 3 organelles are very much important for the cell division. So next, back to the steps of the karyokinesis of mitosis. First phase, that is prophase. In prophase, first appearance is the dissolving nuclear membrane. First one is the dissolving nuclear membrane or nuclear membrane. Get disappear. Okay, this is the first. Secondly, the chromosome become less condensed, or the condensation of chromosome condensation of chromosomes get loosened. In previous class, we have already discussed that the chromosome are the very much supercoiled and condensed form of DNA and histone protein. So this condensation or coiling of the chromosome become decoiled. Okay. Second. Third, the centriole in case of plant cell, in case of animal cell, the centriole get moved towards the two poles. These are the poles and they move towards the poles. Okay, so these are the main changes occurs in prophase. Now in the metaphase. In metaphase, the nuclear membrane completely disappears. Now, metaphase. In metaphase, the nuclear membrane completely disappear and the chromosomes are scattered in the cytoplasm and they get arranged into the equatorial region. How? Like this. Okay, in this equatorial region they get arranged and the centriole of the cell form a fiber like structure which is called spindle fiber. Okay, so in case of animal cell, the centriole or the centrosome form the spindle fiber, but in case of plant, this is formed by the spindle fiber is formed by the microtubule. This is spindle fiber, but this is formed by the microtubule. Okay, but in case of animal, this is by centriole. Okay, and this spindle fiber get attached into the portion. In previous class, I have already mentioned that in the special protein which is get attached in the centromere region, which is known as kinetochore. This spindle fiber get attached in this kinetochore. Okay, so this is the metaphase phase, and this arrangement of this chromosome is known as metaphase plate. Okay, so this is the phase of metaphase. Next one is the anaphase, and this anaphase stage is further divided into two parts, that is early anaphase and late anaphase. It is anaphase. Anaphase is further divided into early and late. In early anaphase, these sister chromatids are detached from each other okay, get detached from each other and travel towards the two pole in the early anaphase. But in late anaphase, there are observed a furrow or a cleavage in the equatorial region, and the chromosome are reached into the two poles. First. Secondly, the nuclear membrane get reappear. Okay, so in late anaphase, in early anaphase, first one, the sister chromatids starts moving towards the pole first. Secondly, in late anaphase, a furrow or cleavage observed in equatorial region. Of course, this is specially for the animal cell. Okay, and nuclear membrane get reappear. Okay, this is the anaphase. Now the last one, that is telophase, which is exactly opposite of the prophase. That is telophase. In telophase, the furrow is clearly observed. Observe the furrow. Okay. Next, the nuclear membrane get reappear. Okay, and other cell organelles get divided into the two poles. Okay, so this is the telophase. Telophase is exactly opposite of prophase. Okay, so this is the telophase. But now question is, the cell still in attached condition, and this is a single cell where two nucleus are present. So it need to be divided into two cells. So that is happened in the cytokinesis or division of the cytoplasm. That is happen in the cytokinesis that is division of cytoplasm. So what happens in cytokinesis? In cytokinesis, in case of animal cell, from this to okay, so this is the two cells, daughter cells. So this is the function. I have drawn that in mitosis, that is, this is the cell and it get divided into two cells, but the process is very complex and it takes time. So this is the process of mitosis or simple cell division, and from this phase the chromosomal material become less dense and scattered into the nucleoplasm. So this is the cytokinesis of animal. But in case of plant, they do not follow or this phase with the animal, like producing a furrow, because they have the cell wall. So in case of plant, the cell is divided by producing cell plate. This is the cell plate. By this, it is divided into two new plant cell. Okay, so if I draw a comparative study of the mitotic division of plant and animal, like that is cell division in plant and cell division in animal. First, in general, that is the spindle fiber, that is if spindle fiber formation. The spindle fiber is formed by the centriole, but in case of, the spindle fiber is get formed in case of plant, that is microtubule, and in case of animal, this is formed by the centriole. Okay. Number 2, that is the cytokinesis. In cytokinesis, it is formed by the furrow, but producing here cell plate. Okay, these are the main divisions, but further the division of plant and animal cell division will discuss later. Now the question is, the cell is divided and new cell is formed. Okay, that's fine. But how these new cells are get matured and give rise another new cells, and if there have so many steps, that is in prophase, metaphase, anaphase, telophase, so if it miss any kind of phase, so what happens? That's why a concept that is called cell cycle. Cell cycle is nothing but a mother cell divided into two daughter cell in mitosis. These two daughter cells get another two daughter cell. Then for this is the mother cell. So from one daughter cell become a mother cell and give rise to daughter cells. This cycle is known as cell cycle. And this cell cycle is divided into two phases mainly. One is interphase, the cell cycle. One is interphase. Another one is mitotic phase. Okay. So in interphase there are three phases, that is G1 or growth 1 phase. Number 2 is S phase or synthetic phase. Number 3 is G2 or growth 2 phase. So what are they and what happens in this? So this is the cell cycle, that is the whole interphase stage, that is divided into three parts, that is G1, S and G2. And this cycle is cycled by this process, that is first G1, then S, then G2. So in G1 phase mainly the changes, that is the protein, amino acid, ribosome synthesis occurs in G1 phase, mainly protein and amino acids. In S phase, here actually the replication of DNA, that is duplication of DNA from one DNA. From one DNA, it produces its replica, that is 2 DNA. Okay. So this is the replication. Now, second is the doubling of chromosome. Of course, when the DNA get replicate and make its copies, the chromosome also be duplicated and make twice. So this is happens in S phase. Now the G2 phase. In G2 phase, here also the protein synthesis, as well as stored fats, so that it can pass into the next phase, that is mitosis. This G2 phase also known as pre-mitotic phase. Okay, this G2 phase is also known as pre-mitotic phase because it is happens before the mitosis. So next to the G2 phase, that is mitosis, which is divided into two phases, that is karyokinesis and cytokinesis. This karyokinesis is further divided into 4 phases, that is prophase, metaphase, anaphase and telophase. Okay, next the cytokinesis. Next one phase remains, that is G0 phase, and this is very much important for a cell to mature. And this G0 phase actually the resting phase of a cell. Resting phase of a cell, where totally the cell activity gets stopped and it will only rest and get matured. If a cell get arrested into this phase, that is G0 phase, it will not able to divide. The example in human body, that is the neuron or the cardiac muscle cell. We know that neuron cell or nerve cell cannot divide because these cells are arrested into the G0 phase. They cannot promote to the next phase, that is G1. That's why they are unable to divide. So this is the G0 phase and its significance. So what is the significance all these cell cycle? The cell cycle the significance of the cell cycle. Number 1, to maturation and preparation of a cell before cell division. Okay, number 1. Number 2, division of other cell organelles. The maximum cell organelles are actually get divided into G1 and G2 phase. The cell organelles also divide. Here also cell organelles also get divided. So this is the significance. And last and most importantly, that is to check the error. Okay, this is very much important step. In the cell cycle, there have so many checkpoints. Here, 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 so many checkpoints are there, which will check that all the phases passed before. Suppose the cell passed the G1 phase, okay, and it will next go to the S phase. So the requirement in G0 phase is get checked into the checkpoint which is located just between the G1 and S phase. And this is very much important for a cell division, because if any error happen, the cell immediately stops at the checkpoint and it will not allow to further divide. So this is the importance of cell cycle in cell division. It will check any error and maintain the normal condition as well as normal scenario of the cell cycle. So that's all for the today's video. If you like to get more such informative video, you may subscribe our YouTube channel Eduins, and also please do follow, comment and share our Facebook page Eduins. Also, you may visit our official website Eduins, where you can get our premium version at a very low cost, where you can get unlimited mock test and study materials. So please do visit right now. Thank you.